Hello, this is Project 2 on longevity genes, and it's by myself, Clay Carpenter.
I'm Owen Carpenter.
And I'm Riley Dodd.
All right, so here's the table of contents. First, we'll go into the introduction, then methodology, results, and conclusion.
So for the intro, so for this project, we were given a data set on yeast cells and the genes that have been found in the yeast cells. So we implemented clustering algorithms to process the different genes within the yeast cells. And we started off by normalizing the given data as a base starting point for the clustering algorithms. And that data that we normalized was the SCH9/WT, the RAS2/WT, and the TOR1/WT. And then based off of the use of the clustering algorithms, we were able to sort the genes into clusters based off of similar characteristics. And by forming these groups, we can make conclusions on whether a certain gene has a low or high potential for being a longevity gene. And for the data normalization, we just used min-max data normalization for re-registering the values of a given data point between 0 and 1, and this is the code that we use to do that. So for the clustering algorithms that we chose, we did k-means clustering, which is virtually just a centroid-based algorithm that groups objects based off of similar characteristics. And we tested the data set using several different k values, and the results will be shown here within the next couple of slides. And then the second one we chose to do was the fuzzy c-means clustering. And that virtually just works by partitioning the data into k clusters based on degree of membership of each data point in each cluster. And then it assigns a membership coefficient to each point in the range 0 to 1, which represents the degree of belonging to a particular cluster. And those results will also be shown here in the next couple slides.
All right, so here we have the k-means clustering algorithm code that we used. First, we have the create centroids, which randomly takes a NumPy choice for creating a centroid inside the dataset. And then we're going to append the centroids and then return them for assigning the clusters. So we're passing centroids in the dataset. And here we have an assign array. We're going to loop through the length of the dataset and figure the Euclidean distance for each centroid to the cluster data point, and then assign that data point to each cluster as here, and then return that. Then we have to update the centroids based on each assignment. So this means that we have to take the new mean for the centroid from each cluster's data points. And so we do this until it does not change anymore. And here's the code to do that. And inside the k-means function, we normalize the data. This isn't needed here because we have it in main, but here we create the centroids, and then we're going to loop while it's true to assign the clusters and update those centroids until it converges where the centroids don't change values. So the new centroids will equal the centroids, and then we're going to return the centroids and the clusters for further evaluation. Next, we have the fuzzy c-means clustering algorithm. Here we have the membership weight matrix. So this is making a membership weight for each data point, and then we're going to return the weights using the Dirichlet formula. Then we're going to compute the centroids. So we're going to raise the weight by a power of m, which is the fuzziness parameter. And then we're going to calculate the sum of the data points for each cluster using this. And then finally, we're going to normalize the sum of the weights for each cluster to get the centroid and then return the centroids. And then we have to update the membership weights based on each centroid, of course. So we're going to loop through the samples and then find the Euclidean distance and then iterate over each cluster for the distance. And then this is the formula we used in class. So I just represented it here to find that. And then we're going to return the weights. And then finally, we have the fuzzy c-means function. We're going to create the weights using the membership weight function, loop while it's true, and then create or compute the centroids. update the membership weights, and then check if it converged. And if it converged, we're going to return centroids and clusters.
All right, and then for results, here are the genes that we found to be possible longevity genes from the candidate genes. Here's the list. I'm not gonna say every single one, but there it is. And then these are possible longevity genes because these candidate genes were found to be within clusters with larger amounts of known longevity genes, which makes them share characteristics with those types of genes.
All right, here's the code that we created for this to work. So we're going to pass the data set, the known longevity genes and candidate genes from the text file provided in the project 2. And so here we're going to loop through the data, checking the gene that is in the k-means cluster, and we're going to get the average count, which is taking the mean, and then furthermore, we're going to take the target cluster and check about if it's the longevity cluster is greater than the average count. And then we're going to check the candidate. If the candidate is in a cluster, we're going to see if it's in k-means, and then we're going to return that candidate and cluster if it is above average with known longevity genes. Also, here's some graphs that we made for the k-means and fuzzy c-means clustering algorithm comparisons. On the left, we're using a k value of 3. And so with k-means, the boundary is a lot more stricter than fuzzy c-means because the fuzzy parameter was 2, so m equals 2. You can see that the boundaries overlap one another compared to k-means. This is the same for k value equals 5 for k-means. The boundaries are stricter than fuzzy c-means. But you can also see in fuzzy c-means, some of the outliers are not their they're not their own clusters or centroids. They're actually based off the central clusters. But using k-means, because the boundaries are more strict, the outliers like the green cluster and the blue cluster are their own separate centroid clusters. Furthermore, k value equals 7, we can see the same things as I just said. Some outliers in k-means are their own cluster. And then the fuzzy c-means looks like a jumbled mess because the boundaries are not as strict as k-means. And this is the same thing for k value equals 9. All right, and here's our conclusion.
All right. So with the conclusion, k-means clustering has stricter boundaries, as seen in the graphs, between each cluster, making characteristics more distinct between each gene. And then fuzzy c-means clustering has a fuzziness parameter of m equals 2, making the cluster boundaries not as strict, allowing for genes to share more outgoing characteristics like the outliers shown in the graph. So what makes a possible longevity gene? Genes that are in a cluster with a dominant amount of longevity genes have a higher chance to be a possible longevity gene. So k-means clustering is more practical at creating strong boundary to find the correct correlation between candidate and longevity genes compared to fuzzy c-means, and this will end up with the most possible longevity genes.
All right, and that's it. Thank you for watching.